Dear students, we are going to study about the nervous tissue today. Nervous tissue, basically the histology of the nervous tissue. As you all guys know that there are various kinds of tissues present in our body. There are muscles, there are bones, there are the connective tissue that make up different components, and all of them they collaborate together and put forth various the functional abilities and various structural features that we that allow us to exist in the nature. So the nervous tissue is basically a very important structure in our bodies, both anatomically and physiologically, and that enables us to perform not only to perform but the support life, but to support life as well. So let's dig into it, what it is and what it is comprised of. So it basically primarily does the communication and the control system. The entire structures, the various structures in the body, they communicate through the nervous system, and they're controlled by the nervous system as well. And obviously the nervous system is formed up of the nervous tissue, and it can be divided into the structural categories and into the functional categories. The nervous system, as you all know, that it could be divided into the central nervous system and the peripheral nervous system, and then there's another nervous system which is called the autonomic nervous system. Here we'll just touch on to it. The central nervous system, you know that we have the brain and the spinal cord, whereas in the peripheral nervous system you have cranial nerve, you have spinal nerves, and the ganglia. And when you will combine the ganglia with the spinal nerves, or even with the cranial nerves, they form part of the autonomic nervous system. Rather, both the central nervous system and the peripheral nervous system together, they constitute the autonomic nervous system as well. So the functional components of the nervous system, they are the sensory afferent divisions and the motor efferent divisions. You know what the afferent division that receives sensory information from the receptor and transmits information to the central nervous system, whereas the efferent division transmits motor impulses from the central nervous system to muscles or the glands. So in the sensory division, it has two components: the somatic sensory components. Then in the somatic sensory, there are general somatic senses, the special senses. Otherwise, there are other two, the visceral sensory components, and then there are the motor division. It also has two components. It's just I'm just touching these aspects because they're basically related to the general anatomy, not your histology, but for your information. The motor division has two components as well: the somatic motor and the autonomic motor system. Now, from here, we will talk about the components, the structural and the functional units of all the nervous systems. The entire nervous system is composed of a simple body, which are called as the nerve cells. And the nerve cells, they are basically divided into two: the neurons and the glial cells. The neurons, they are the excitable cells. They initiate and transmit the nerve impulses, whereas the glial cells, they are the non-excitable cells, and they support and protect the neuron. So there are two types of cells basically within the nervous tissue. First ones, they will transmit and communicate and control and collaborate with the nervous impulses, with the neural impulses that are being generated within our body. And then the glial cells, they are going to support these neuronal cells for the nutrition, for the support. Now let's talk about the characteristics of the neurons. The neurons, they have a high metabolic rate. They have extreme longevity, and they are typically non-mitotic. Let's talk about their structures. The neurons or the nerve cells, they form the structural and the functional unit of the nervous system, and there are almost 10 billion neurons in the human nervous system. Although they show the greatest variety of sizes and shape of any group of the cells in the body, they fall into three general categories. There are sensory neurons, there are motor neurons, and then there are the interneurons. All neurons have a cell body and the processes, the axons and the dendrites. The cell body of a neuron contains the nucleus and those organelles that maintain the nerve cell. The processes extending from the cell body constitute the single common structural characteristics of all the neurons. Most neurons have only one axon, usually the longest process. Some axons may be more than 1 meter long. A neuron has many dendrite processes that are shorter and usually thicker than an axon. The neurons are classified on the basis of the number of processes extending from the cell body. Thus, they can be multipolar, they can be bipolar, they can be unipolar. The multipolar, they have one axon, two or more dendrites. The bipolar ones have one axon and one dendrite, whereas the unipolar ones, they have one process, the axon, that divides close to the cell body into two long processes. The motor neurons and the interneurons are multipolar. In interneurons, which constitutes most of the neurons in the nervous system, the physiologic direction of impulses is from dendrite to cell body to axon or from cell body to the axon. Thus, functionally, the dendrites in the cell body of the multipolar neurons are the receptor portions of the cell and their plasma membrane specifically adapted for impulse generation. Axon is the conducting portion of the cell and its plasma membrane is specialized for impulse conduction. The terminal portion of the axon, the transmitter or effector portion of the nerve cell, contains various transmitter substances that affect other nerve cells as well as muscles and glands. The cell body is also known as soma or the perikaryon. So the sensory neurons, they are unipolar, whereas the true bipolar neurons, they are limited to the retina and the ganglia of the vestibulocochlear nerve, the cranial nerve 8. The cell body of the neuron has the characteristic of a protein secreting cell. Now this is interesting. For that, you should know what are the protein secreting cells and the mucus secreting cells. The cell body of perikaryon is large, dilated region of the cell, contains large euchromatic nucleus with a prominent nucleolus. Nerve cell pericardia contain inclusions, the nasal bodies, that stain intensely with the basic dyes and metachromatically with thioenine dyes. Each nasal body corresponds to a stack of rough endoplasmic reticulum. The perikaryon also contains numerous mitochondria, large perinuclear Golgi apparatus, lysosomes, microtubules, neurofilaments, vesicles, and inclusions. Nasal bodies, free ribosome, occasionally the Golgi complex, extends into the dendrites but not into the axons. In both light microscope and transmission electron microscopes, this distinction helps us to distinguish between the axons and the dendrites. The combination of euchromatic nucleus, large nucleolus, prominent Golgi apparatus, and nasal substance is indicative of the high level of anabolic activity needed to maintain these large postmitotic cells. The neurons, they do not divide. They must last for a lifetime. They do not replicate. The subcellular components of the neuron turn over regularly, having molecular lifespans measured in hours, days, and weeks. The constant speed to replace enzyme transmitter substances, membrane components, and other complex molecules explains the presence of morphological features characteristic of a high level of synthetic activity. Newly synthesized molecules also need to be transported to distinct locations. What about the dendrites and the axons? The axons, they have branches which are called as collaterals. The axon terminals are the telodendria, and then they end up into the synaptic knobs. We all have talked about these. Now let's see. The dendrites, they are normal processes that receive stimuli from other nerve cells or from the environment. They are located in the vicinity of the cell bodies. They have a greater diameter as compared to the axons. They are unmyelinated. They are usually tapered and form extensive arborizations, which are called as the dendritic trees. Dendritic tree significantly increases the receptor surface of a neuron. Many neuron types are characterized by the extent and shape of their dendritic trees. There is generally no sharp delineation between the contents of the cell body and those of the dendrites, whereas the Golgi network remains close to the nucleus. Other organelles characteristic of the cell body proper, including ribosomes and rough endoplasmic reticulum, are found in the dendrites, especially in the base of the dendrites. What about the axons? They're larger, typically longer nerve cell processes. They extend from the cell body, and the portion of the axon which extends or which comes out of the cell body is called as axon hillock. They are also called as nerve fiber, and most neurons have only one axon. So The axon can be extremely long. Axons that originate from neurons in the motor nuclei of the central nervous system, which are also known as the Golgi type 1 neurons, they travel more than a meter to reach their effector targets. For example, the skeletal muscles. Interneurons of the central nervous systems, which are known as the Golgi type 2 neurons, in contrast, have a shorter axon. Although an axon may give rise to a recurrent branch near the cell body, that is the one that turns back towards the cell body, and to the other collateral branches, the branching of the axon is most extensive in the vicinity of the targets. The axon originates from a conical projection of the cell body that I have already told you is the axon hillock. It is basically devoid of the large cytoplasmic organelles such as the nestle bodies and the Golgi cisterns. Microtubules, neurofilaments, mitochondria, and vesicles, however, pass through the axon hillock into the axon. It is here that molecules synthesized in the cell body and destined for transport to distant portions of the neuron enter the axonal transport system. These systems, I will talk about those in a minute. The region of the axon between the apex of the axon hillock and the beginning of the myelin sheath is called the initial segment. The initial segment is the site at which an action potential is generated. The action potential is stimulated by impulses carried to the axon hillock in the membrane of the cell body after other impulses are received on the dendrites of the cell body itself. Axons in the central nervous system may be insulated by a myelin sheath, especially in the tracts that constitute the white matter, or may be bare unmyelinated, as in the integrative portions that constitute the gray matter. Axons in the peripheral nervous system, they are never completely bare, although they are described as myelinated or unmyelinated. Even the unmyelinated axons are embedded in the imaginations of the Schwann cells. So the axon, they end up in telodendria and the synaptic knobs, which are also called as the terminal butons. The axon transmits a nerve impulse away from the cell body towards another cell, but that totally depends how the cell or the neuron is being oriented. I have already talked about all these structures. The cytoskeleton within the neuronal cell body. Here is a typical neuron. Here is a histological view. And here is the classification. I have already talked about it. Morphological classification, the structure and function classification. Structural classification, you have unipolar, bipolar, and multipolars. There you go. The functional classifications, they are sensory efferent, motor efferents, and the interneurons. So the interneurons are the association neurons. They lie entirely within the central nervous system and are multipolar. They receive impulses away from many other neurons. They carry out the integrative functions of the nervous system, and they facilitate communication between sensory and the motor neurons. So what are the glial cells? You already know that the supporting cells of the nervous tissue, it means the nervous tissue or the neurons, they form the structure and the functional unit, but they have to be supported. For what? For nutrition, for their conduction purposes. So the cells that are supporting the nervous tissue itself are called as the glial cells. They're also called as the neuroglia. And they occur within both the central nervous system and the peripheral nervous system. Obviously, they are smaller in size as compared to the neurons, and they are capable of mitosis, that is, they can be divided. And they are unable for the conduction purposes. They physically protect the neurons, they nourish the neurons, they supply or provide a supporting framework. The glial cells, they far outnumber the neurons, that is, they are greater in number as compared to the neurons. And they account for about half of the volume of the nervous tissue. There are different cells, we'll talk about them. You can see the oligodendrocytes, you can see the microglia, you can see the astrocytes, you can see the ependymal cells, the small apical cilia, and you can see the myelin sheath as well, the myelinated axons. We're going to talk about it as well. There you go. You can see an astrocyte. And then again, the ependymal cells. The ependymal cells, the specialized cuboidal cells with the apical microvilli, and they line the ventricles of the brain. Now, first and foremost, when you're going to talk about the neuroglia, you say that within the central nervous system, the supporting cells are designated into three types. They can be oligodendrocytes, they can be astrocytes, and they can be microglia. The oligodendrocytes are the myelin-forming cells of the central nervous system. The astrocytes are the cells that provide physical and metabolic support for the neurons of the central nervous system, whereas the microglia are the phagocytic cells of the central nervous system. So let's first talk about the astrocytes. They exhibit a star-like shape due to the projections from their surface in the most abundant glial cells. They constitute over 90% of the cells in some areas of the brain. They help form the blood-brain barrier. You should know about what blood-brain barrier is and the blood CSF barrier is. They are actually the gateways. They are actually the control channels. They basically sort out what is going to enter into your brain and what is not. They are the main filters, what is going to go out of your brain and what is going to come in. This is the site which controls it, and there are 5 regions in the brain that are there that lack the blood-brain barrier. I can give you example, but let's leave it to some other point. So they regulate the tissue fluid composition, they provide structural support, they replace damaged neurons, and they assist neuronal development. What else do they do? They can be classified into protoplasmic astrocytes and the fibrous astrocyte. The protoplasmic ones, they are more prevalent in the gray matter. The fibrous astrocytes are more common in the white matter. The both types of astrocyte contain prominent bundles of intermediate filaments composed of glial fibrillary acidic proteins. The filaments are much more numerous in the fibrous astrocytes. Antibodies to GFAPs, they are used as specific stains for astrocytes. They have elaborate processes extend between the blood vessels and neurons. The ends of these processes expand and they form end feet that may cover large areas of the outer surface of blood vessel or of the axonema. That's how they are going to make the blood-brain barrier. Astrocytes play a role in the movement of the metabolites and waste to and from the neurons and regulate ionic concentrations in the intercellular compartments. Hence, they maintain the microenvironment of the neurons. They regulate the function of the tight junctions of the brain capillaries and venules. Astrocytes also provide a covering for the bare areas of myelinated axons, that is at the node of Ranviers and at synapses. Perisynaptic astrocytes may confine neurotransmitters to the synaptic cleft and may remove excess neurotransmitters by pinocytosis. Now, here we have another type of cells which are called as the ependymal cells. I've already told you that this is a cuboidal epithelium which with the cilia on the apical surfaces. It circulates the cerebrospinal fluid. They line the internal cavities. What are the internal cavities? They are basically the ventricles. The processes make contact with other glial cells. They help form the choroid plexus, the cerebrospinal fluid, CSF means. So, let's talk about the microglial cells. So the microglial, they constitute the smallest percentage of the central nervous system. They are phagocytic. Whenever there is an infection, they go to that site, they remove the debris, and they function more like the macrophages. Their characteristic feature is that they are present in very small number, but they can proliferate and they become actively phagocytic in regions of injury and disease. They are considered part of the mononuclear phagocytic system and are believed to originate in the bone marrow, and then they enter the CNS from the blood. There is also evidence they remove debris during the development of the nervous tissue. They are the smallest neuroglia cells in terms of their size, and they relatively have smaller nuclei when stained with heavy metals. Microglia exhibit short, twisted processes. Both the process and cell body, they are covered with numerous spikes. The spike may be equivalent of the ruffled border seen on other phagocytic cells. There is a little rough endoplasmic reticulum there, and there are few microtubules or microfilaments in them as well. Now let's talk about the oligodendrocytes. They're very important, and they're very important in terms of how they are involved in the myelination process. They're large, big body and processes, and they form myelin sheaths and they speed up the transmission. How the myelin sheath, the speed up transmission, all of you must have been aware of these electrical wires all over your within your home appliances. You must have seen one. All of the electrical wires, they carry current and they have been insulated, all of them. What is this insulation and why do we have it? We want it so that the electrical current can flow easily and it shouldn't dissipate. That's the same reason, or that's the same philosophy that has been carried from the central nervous system, basically, or the neuron is a conducting cell, and that electrical current that flows through it has to be insulated. So the transmission of the impulse is more efficient and smooth, and with lesser or least, you can say, dispersion. So oligodendrocytes, they produce the central nervous system myelin. The myelin sheath is formed by concentric layers of oligodendrocytes plasma membrane. Formation of this sheath is more complex, however, than the simple wrapping that occurs in the peripheral nervous system. The oligodendrocytes appear in specially stained preparations as small cells with relatively few processes compared with the astrocytes. They are often aligned in rows between the axons. Each oligodendrocyte gives off several tongue-like processes that find their way to the axons, where each process wraps itself around a portion of the axon, forming an internodal segment of the myelin. The internodal myelin segments formed by the multiple processes of a single oligodendrocytes may be on an axon or on several nearby axons. The nucleus containing region of the oligodendrocyte may be at some distance from the axon it is myelinating. What happens actually is that the plasma membrane of the oligodendrocyte become concentrically wrapped around a portion of the neuron. Because a single oligodendrocyte myelinates several nearby axons simultaneously, the cells cannot spiral around each axon in an independent manner, as the Schwann cell does in the peripheral nervous system. Instead, each tongue-like process appears to spiral around the axon, always staying in proximity to it until the myelin sheath is formed. Thus, peripheral nervous system myelin may be described as forming centrifugally by the movement of the Schwann cell around the outer surface of the newly formed myelin. The central nervous system nerve myelin, on the other hand, may be described as forming centripetally by the continued insinuation of the leading edge of the growing process between the inner surface of the newly formed myelin and the axon. There are several other important differences between the myelin sheath in the central nervous system and the peripheral nervous system. In the central nervous system, it exhibits fewer Schmidt-Lehmann clefts because the astrocytes provide metabolic support for the central nervous system neurons. Unlike the Schwann cells of the peripheral nervous system, the oligodendrocyte does not have a basal lamina. Thus, where myelin sheaths of adjacent axon touch, they may share an intraperiod line. Further, because of the manner in which oligodendrocytes form central nervous system myelin, there may be little or no cytoplasm in the outermost layer of the myelin sheath, allowing the myelin of adjacent axon to come into contact. Finally, the nodes of Ranvier in the central nervous system are larger than those in the peripheral nervous system. There is more exposed exolemma, thus making saltatory conduction even more efficient in the central nervous system. Another difference between the central and the peripheral nervous system in regard to the relationship between supporting cells and neurons is that unmyelinated neurons in the central nervous system are truly bare. They are not embedded in the glial cell processes. The bare unmyelinated axon, as well as the absence of basal lamina material and connective tissue within the substance of the central nervous system, help to distinguish the central nervous system from the peripheral nervous system histological sections. And so let's talk about the other cells as well. The other cells are the Schwann cells and the satellite cells. The satellite cells, they're flattened, they cover somas in ganglia, and they separate somas from the surrounding tissue fluid, and they regulate the exchange. Why they are called as satellite cells? So the neuronal cell bodies of ganglia, they are surrounded by a layer of small cuboidal cells that are called as the satellite cells. They form a complete layer around the cell body. Only their nuclei are visible in hematoxylin eosin preparations. In paravertebral and peripheral ganglia, neuronal cell processes must penetrate between the satellite cells to establish a synapse. Like Schwann cells, satellite cells also originate from the neural crystals. They help to establish and maintain a controlled microenvironment around the neuronal cell body in the ganglion. What about the neurolamocytes or the Schwann cells? They does the myelination in the peripheral nervous system. Myelination, I have talked about it and I have told you the differences between the peripheral nervous system myelin sheath and the central nervous system myelin sheath. So, what you have to know about the myelination process is a process by which an axon is wrapped within a myelin sheath. Oligodendrocytes makes it within the central nervous system, peripheral nervous system, Schwann cells does it. It's a protective fatty covering. It has a glossy white appearance. It protects, insulates, and supports the axon. I've already talked about it. This is the process. This is how the two cells are doing it. Neurolamocyte. The two differences between the unmyelinated axons and the myelinated ones. In the myelinated axons, the nerve impulse jumps from neurofibril node to the neurofibril node. It's called saltatory conduction. It requires less energy, whereas in unmyelinated, it travels the entire length of the axon. It's called continuous conduction and it takes longer. So in the central nervous system, almost there is no regeneration, but there are some researchers that say that regeneration is possible in the peripheral nervous systems because they are vulnerable to cuts and trauma. It can regenerate if some neurolema remains. And it depends on three factors: amount of damage, secretion of the nerve growth factors, distance between the site of the damaged axon and the efferent organ. Then this if you want to know, is for your interest, although I don't think there is a need to go into it, but you can read it on your own if you have interest into it. So how a nerve is formed? A nerve is like a cable, most definitely, where the axons are arranged parallel in the form of bundles. There are three connective tissue wrappings: the endoneurium, the perineurium, and the epineurium. The endoneurium is the delicate layer of loose connective tissue, is the innermost layer. Then and this endoneurium is basically formed around the axons, and there's one axon bundle basically, and then various axon bundles, they are gathered together and within a bundle, and that bundle is surrounded by a perineurium. Endoneurium is actually around a single axon. So all the axons, they get together in a bundle and then they are surrounded by a perineurium. Then various bundles surrounded by the perineurium, they are arranged within a single bunch that is called as the nerve, and that is surrounded by the epineurium. It is a dense, irregular, fibrous connective tissue. It encloses the entire nerve, provides support and protection. There you go. So an axon, you can see over here that the axon covered by the myelin sheath, and myelin sheath is furthermore covered by the endoneurium. And then they are forming the fascicles, which are covered by the perineurium. And what is the epineurium? You can see over here. There you go. So the synapses, they can be presynaptic, they can be postsynaptic. Neurons, basically. Then the types of synapses can be exodendritic, exosomatic, exoaxonic. There are electrical synapses, which are the gap junctions, the chemical synapses, which use the neurotransmitters. In humans, synapses occur primarily between smooth muscle cells. Quicker, uniform innervation is essential, all or none principle, as you all know. And the electrical synapses are also located in the cardiac muscle. A very important point. The chemical synapses, they facilitate interactions between neurons, between neuron effectors, cell junctions, the presynaptic membrane, and the postsynaptic membrane. So there are neurotransmitters, basically the chemicals that interact, that helps the presynaptic neuron and the postsynaptic neuron to interact, convey the message from the presynaptic neuron to the postsynaptic neuron. Then it is talking about the neuronal circuit or pathways. They are basically different patterned groups that are called as neuronal pools, and they are defined based upon function, not anatomy. And there are 4 types of circuits: converging, diverging, reverberating, parallel after discharging. Here are those circuits. It's for your interest as well. So this is the end of the lecture. I hope you have understood it. If you have any questions, you can post it, and I wish there are a few points that I can explain to you in a little bit more detail. Let's see, we'll make another video, or you can post me your questions. You're welcome. Thank you so much.